Okay, so I mean, I'm interested in the forecasting from the ecological side, but I don't claim to be that much expertise in that, but give me time. Okay, so anyway, okay, I'll try to run through why we're interested in possibly ecological forecasting for pollen predictions. Because as far as pollen is concerned, with at the intersect of meteorological forecasting, and in addition to what's happening with landscapes, so gets to be very, very big area. Okay, and then I acknowledge contributions from past and current postdocs, including Ha Nguyen, Xiaoyun Chi, and Rakesh Devadas. Okay, it's working. Okay, so just to introduce the aerobiome. So basically, we don't see it, but we're walking through this all the time. And whether we like it or not, we're basically connected to our environment and the landscapes that surround us, both inside, whether you're in urban areas or in peri-urban areas or from rural landscapes. So these spores, whether you're talking about pollen or fungal spores, obviously they connect to ecology since it has to do with reproductive flowering cycles or decomposition of soil organic matter or litter and so on. And okay, the thing is that why is this stuff getting worse? So all of these bioaerosols, these are the approximate sizes. And that's so in relationship because currently air quality monitoring is not so much bio, but more chemical. And that's important too, right? The dust and everything else. Okay, so this is just to kind of give the applications of this. This is a study by Jeremy Silver, who worked down near Melbourne on the thunderstorm asthma project. And he gathered hospital admission data to study what is the prevalence on a seasonal basis of asthma hospital admissions. And anytime I see the word seasonal, I kind of get excited because that's what I like to try to focus with landscapes. So he's got a nice set of 4 peaks here for a total population. And he did divide this by all types of gender and population classes, but I didn't print all that out. So, so these nice 4 peaks, the first one is basically back to school time. So that's an interesting period where you get peaks in a hospital. The next 2 more or less are sort of like winter related peaks. And then of interest to us would be the last peak in November. That is strongly related to grass pollen, which coincides exactly with the pollen season in Melbourne. And it's kind of cool because when you go through the different age classes, you see some of these peaks disappearing, like, back to school. If you happen to be over 30 or something, you're not really going back to school anymore and stuff like that. So. Okay, and then there is this nice figure that if we go back to the 1960s, there is this documented more or less increase in asthma activity and allergic symptoms and that. So it's kind of got a lot of attention as to why we're seeing this increase. Okay, so here in Australia, it's not that easy to study pollen or develop good forecasts from a few perspectives. One is it's pretty much a young science for Australia, but another part is that we only have very sparse sampling of pollen in the air from like this Burkhardt spore traps that tend to be only present in the capital cities, more or less, and you tend to see one available for that entire part of the territory. So it's very hard to do forecasting other than if you're working at the very site-specific scale. I mean, on the good hand, you can say that these are placed, if not inside, very close to urban areas, so they would affect a greater population. But from forecasting, we need to look at more than just how it works at a particular city. And we'd like to be able to go cross-continental, as being done in Europe and attempted now in North America. So a little history, because it's been about 10 years now. In 2013, Janet Davis put in an ACES proposal that got funded. And in that proposal, she convened for the first time the operators of pollen stations throughout Australia and New Zealand. So in many instances, these operators had been working basically solo and rarely have even known each other. So it was, to me, it was the beginning of putting together an Australian forecasting system, the very, very beginning, the spark. A highly interdisciplinary group from ecologists on to health scientists and biostatisticians and so on. And this group was so popular that we met twice in 2013 and 2014, each time for 1 week long workshops at Stratford Island in Queensland. And it was also a very productive group. Just about every person in that workshop each led a paper. And by the time the second workshop convened in 2014, this first paper had already just come out in Plus One by Simon Haberley in Canberra at ANU. And basically he did a comprehensive analysis of the pollen composition and the macroecology of airborne pollen across these sites. So I guess just to remind you, when this group met, it was the first time people were able to look at pollen across different sites across Australia and New Zealand. So for the first time, we actually got a chance to mix data sets and play around with climate, geography, and so on. And just for we know, the number one pollen was from the Poaceae family, or grasses, but along with these, I think it was the top 10 or 20 that he showed off in the paper. And then by the time the second workshop had ended, Janet Davis led a transdisciplinary manuscript that kind of connected all of the different cross disciplines present at the workshops. So we're talking about from the health scientists to pollen dispersion models, to diversity, phenology, plant biology, and biogeography. So it was a very nice, excellent paper tying in everything from the biogeography, landscapes, all the way down to the human lungs. And there's just two other papers I wanted to note, even though I guess there were a lot more than this. I thought that Daniel Meddick's paper was a very nice tie-in to what work I've been doing and so on, and that she put together, like from the Atlas of Living Australia, a map of C3 and C4 grass species. So she actually dug in and tried to get as much detail, so beyond Poaceae or beyond the family level, but she tried to dig as much as possible to put, just to get a sense of the biogeography involved in what we're dealing with, since C4 grasses in particular were highly allergenic, whereas the C3 species, of course, were allergenic from the point of view of being the exotic species. So like ryegrass. So all of this is very important for the whole group in particular, but I thought it was an excellent paper to put all this together like this, and pretty much replicated what meteorological models had predicted for C3, C4 composition. Just so you're aware, because the colors aren't the best, the darkest black here is solely C3 grass species, whereas the shaded gray color here, or dark gray, is the mixed C4 and C3. And then the light gray colors are C4. And either there was no data available or there were no grasses in the other parts there. And of course, the last one, because of the nature of this talk, is my lab then led this paper that had to do with being, literally, we thought we were the first because no other satellite-based remote sensing study had been published at the time, but the lab in the UK actually came out with a paper before us. But of course, not to our knowledge until this was done. So, but anyway, this was connecting remote sensing, looking at land cover types and so on, and starting to look at land surface phenology, LSP, and connecting that with the pollen data that was being collected by this group and standardized by this group. Okay, just to recognize the other outputs from ACS workshops, led by Janet Davis again. She went on to lead an NHMRC partnerships project, which spawned the OSPollen network, or OSPollen partnership. Okay, and besides that, from my lab group, we managed to get 2 discovery DP projects based on this workshop, literally, from looking at pollen. So we think it was a very successful ACS group. I'm only going to show you the concluding work from the OSPollen paper, let's say. So this was put together as a synthesis as to what had occurred throughout the 4-year NHMRC project. And what was in this paper is looking at the 4 main sites from Brisbane, Canberra, Sydney, and Melbourne, and tying all of that information together with respect to meteorology, biology, and all these factors, and looking at this for a good 4-year, what is considered very high quality data set. Okay, and to this day now we can go into a pollen site where we have pollen forecasts available. So I went in today. Again, it's okay, and it's basically the best that's being done right now. But this is an example, like today would be the value low. So, but again, this is the beginning. We're hoping that with better modeling and so on, that we're headed towards more sophisticated forecasts. Okay, so I wanted to go from the landscape point of view, some of the main sections that we believe that we can contribute to pollen forecasts. So one is that literally all pollen emissions and sources come from the landscape. So I think we have the right to be included then. in looking at what we know about landscapes and add our expertise and contribute our expertise about landscapes. So, the first and foremost is you at least want to have a map that you can use as your background matrix from which you can apply all kinds of methods for pollen forecasting. So we want to map the sources. In the Devadas paper that we had generated, we sort of had chosen the IGBP land cover classification, which is probably as coarse as you can get because there's only 16 classes available and one being grasslands. The bad news about that was that if we go to a very high pollen producing region like around Canberra, we would see that from IGBP perspective, there is no grass to worry about. But of course, we know that within the classes that are available, there is grass present. It's just the way the nature of the classification doesn't make it easy for people studying pollen to extract the information that they want. You can go more national. We did the IGBP because our study involved France and the US, since part of the ACS group was the director of the French pollen network. So we can go to ENVIS and get much more detailed groupings of vegetation from which we can probably pull out better, more quantitative estimates of grass cover or grass amount present, but it's still not ideal. In particular, if we look at the eucalypt open woodlands classification, we would see there's a lot of grass in there, but it would be hard to know that or pull that out from just looking at the classifications. And then even then, if you're forced to go and accept this class, are you accepting the entire class? Just some of the problems, I think, in doing this. There's a ALUM is getting even finer resolution, and we use it. It's useful. It includes land use, which is also good to know because pastures are important and so on. I guess I should point out that one of the issues we have with all of those classes is the urban class, since inherently a lot of classifications would like to just call urban, urban. Yet we know that urban has pollen sources. So, okay, so going back to the mapping the sources, this would just be an example of using another land cover data set, Geoscience Australia, the dynamic land cover data set, DLCD. And from all these classes, they've got some really nice grass ones because they have a class called rain-fed pasture. And there's also others called hummock grassland, tussock grassland. So the classification matches more of our purpose. And then this would be the result that we're aiming for. We just want a mask that we can use, meaning that this should be better than using no mask at all, right? So if we want to know where is our sources relative to where our samplers are going to be, then this is a good example of the map. Now we have finer resolution data sets available. And I should have mentioned that the ALUM is generated from Landsat data sets. So they're based on satellite data. It's at 30-meter resolution. And nowadays, the Europeans, with their successful Sentinel mission, have 10-meter resolution land cover maps. And at 10-meter, it's fairly easy for this classification scheme to just say right out the bat, this is tree or this is grass, as well as this is built environment. Because just at 10-meter, you don't worry so much about things mixing, and you're not after what is the dominant species in each 10-meter grid cell. It's easy to just say this grass. So it's a little nice, sophisticated, because it uses our radar information as well as optical methods. Since radar gives estimates of roughness, it greatly enhances the ability to look at the 3D structure of the pixel as well. So this is an example of the Brisbane area, red color being urban and yellow being grass and forest. That's the Sydney region where we can see grass and forest. We could zoom in much closer to individual pollen stations. So Brisbane has these two at Rocklea and Mutapilli. And Sydney actually has three, but these are the two that were financed by the ARC project at Macquarie University, and Richmond at HIE has the pollen station co-located with the air quality station there. So you can see that zooming in, it's fairly easy even for like Macquarie to see the patches of yellow grass here and there. And we see a nice parallel study here with Richmond being peri-urban, a lot of yellow, Macquarie being urban, very little yellow, and by same situation with Rocklea and Mutapilli. So these add some nice background matrices to try to understand why pollen readings may differ or be the same. But they're better than present day, where we're just using one station at Campbelltown to forecast the entire Sydney region, the entire basin. Next thing about the land cover is that you want a methodology that will allow you to rapidly measure land cover changes. So you just don't want to know the class and then assume the class is there forever. So there is massive land cover changes going on, especially in Sydney West. And satellites would be a good way to keep on top of that. And I'm going to show an example from our group on that. So this is a paper by Dr. Xiaoyun Qi, who went ahead and started off with the nice C3, C4 map that Daniel Nedek had produced, but started looking more at the intensive pastures of Eastern Australia, since this is where the vast majority of pollen emission sources are believed to have occurred. And she's investigated this in more detail. And what was nice about that is that she studied the phenology of these areas. So moving up from just land cover to also looking at phenology, and then looking at this for 18 years, so adding a third dimension now, which is time. So looking at phenology within a year, looking at time series across 2 decades, and geographically looking up and down at the emission sources. So she noticed that if she were just, for every pixel there, if she just asked, give me the date at which you are at maximum productivity, your maximum greenness, so to say, for the grasses. Why we want that? Because we believe that when you are at maximum, you're switching from a vegetative phenological processes to reproductive phenological processes. So your energy is now being diverted toward other things, and you're not going to grow anymore. So peak is kind of nice from that perspective. So she plotted histograms of the peaks, and she noticed up in more of the Queensland, southern Queensland area, that all of the peaks of all those pixels seem to fall in the autumn. And then if she went to Sydney region, you would see there's a nice fair amount of peaking in the autumn, but a higher amount peaking in the spring. And you go down to Melbourne and mostly everything is in the spring. So what's important for that is it's a way of separating, if I'm not going to argue it's exact, but it's a way of separating C3 from C4 grasses, cool season from warm season grasses, which adds now, instead of just having grasses, you have type of grass. And we, again, keep in mind that the C4 is more allergenic than the C3. So she did that for every year, saw the interannual variability. The nice thing about this, doing this by satellite, is that every year the C3 could turn off and the C4 be active, or vice versa. The reason for that is you may have drought, cool season drought, or you may have the event drought happening in summertime. So it's like part of the seasons can switch on and off based on ecohydrology, so to say. Okay, and what she found basically was from a decadal time scale from 2003, that C4 grasses are increasing while C3 is decreasing, is her finding. And geographically, she's showing that happening such that there's a migration towards the south. So that's another type of land cover, right? Because land cover change also includes redistribution or shifts, geographical shifts in species composition, which is going to be important for pollen. Okay, switching, that was kind of a nice tie-in that connected both mapping emission sources towards monitoring emission sources. So we can monitor things from phenocams, and we can also monitor things from satellites. And more often than that, we would want to do both. So this is what I was trying to point out about the phenological behavior of a grass, that you're reaching a peak and then you expect that it's switching toward a reproductive stage and then a ripening stage. And now we want to overlay the LSP, or land surface phenology, the red line, over a pollen season, the blue behavior in here. And now, can we connect these two? Because if one leads to the other, then that means you have a powerful first step predictive tool to know that, if you got an early vegetative growing season, can you venture out to say that we're expecting an early pollen season? So again, that's conjecture at this point, but that's what we're asking. I also want to point out that when we are looking at the vegetation, I always like to mention the fact that you're looking at grass dynamics, because our trees just don't do this every year like that. And in there, you can see green waves, meaning that the vegetation starts somewhere in Victoria and shifts to somewhere else in Victoria, and it does that repetitively. In Sydney or in the mid regions, it's much harder to see that wave because you've got the complicated nature of both C3, C4 grasses altering that nice smooth pattern. And so that kind of happens as you go more and more northward. So all we're doing is looking at the EVI here, greenness index, moving along, and underneath it, we're watching the pollen coming up and going down, and hoping that we can make linkages between the two. So I like to mention that green wave because it means that when a pollen station records pollen, it's very possible if it's downwind that it's reading pollen from far away, and there's no flowering anywhere near that pollen trap at that point in time. So this adds part of this complication to this, that we're seeing things Actually, the pollen trap is a sink. It can see lots of things that DNA analysis it can reveal about what is in the landscape that we wouldn't know otherwise. Okay. Okay, so this is a map of the state of Victoria, masked for just the grasses. And in this particular case, we just want to know what is the peak EVI value for this year, 2016, which is a very high attention year since that was the date of the thunderstorm asthma event. And we can do a standard anomaly of that with 20 years of data and see that the peak EVI was at Z-score 2. So unusually high, lush biomass at the time that we had that thunderstorm asthma event. At the same time, I can look at that same peak pixel and say, I don't care about the peak EVI value. I want to know the peak EVI date. When did it peak? And we see here, it's amazingly high in red, just above Melbourne at the time, which means that it peaked in late October, early November. And from an anomaly point of view, that was at least 30 days unusual lag. And we sort of know that when you have a wet year, you have lags in vegetation phenology. And when you have dry years, you tend to have advances in vegetation phenology, since if it's sort of dry, then the vegetation is quick to reproduce, quick to flower, quick to peak. So, anyway, we believe this is interesting information for thunderstorm asthma predictors, because if the peak is later and later in the year, you're entering thunderstorm season. So you have more probability of crisscrossing thunderstorm events right at the wrong time or the wrong or right, I don't know. But you don't want them to intersect with the pollen season too much. Okay, so when we look at these relationships between the land surface phenology and the pollen data, we do get into a complicated situation where they do change by site. So places in Europe, like these 3 French sites, they're monocultures, and it almost look like they coincide with each other. And then when you go to Melbourne, it's can be almost 2 months between the peak in greenness and the peak in pollen. And Sydney seems to be in between. I know we have the double peak to make it harder to decipher, but the Devadas paper showed about 4 weeks shift between the two. So that's something we still need to think about, and whether or not, we just need to worry about. Wow, I didn't know I had that still. All right, so that's just the land cover maps. Okay, so recently 2 other people are following the same line. Andrew Richardson got involved with a group in Spain, and they used the phenocam to look at their grasses, Mediterranean grasses. And it looked like they kind of resembled the pollen shooting up, similar to what is being observed in Melbourne or Victoria. And they have a nice conceptual diagram of separating and then studying the connection between vegetative and reproductive phenology cycles. So what we're trying to do there is is there something like the start of the growing season that could help you predict something about the pollen season, and sort of like combining these two in certain ways. And just one other study that just recently came out would be in Canada, where they were looking at pollen and satellite data as well, or for tree species. So they're just taking the satellite products of start, peak, and so on, and relating that to the pollen profiles. And their estimates or prediction estimates here for these three tree pollen species. So, okay, and then our own work here is we've been looking at some of the pollen patterns for 2 decades or almost 3 decades, and seeing general advancing pollen peak. So in line with the behaviors you're seeing in the Northern Hemisphere, that the warming climate is advancing the pollen season. And we sort of can generate a plot like this to look at the original pollen season in the last 12 weeks of the year and the what it is today. So a shift over. Thought there was another one. Oh, okay, sorry. Okay, so that's similar to what Anderegg found with looking at pollen data in North America. Advancing and a longer season, which means greater pollen exposure and greater chance for sensitization of patients. And same with the ragweed season being reported. So the other part that Ha, our postdoc, did here is also look at the peak of the vegetated grasslands around Victoria. And in that case, green signifies that it's advancing for the last, well, she did it for 3 decades with AVHRR, but it was pretty noisy. So she wound up with a 2-decade MODIS-based study that's more reliable. So the thing that would be interesting here from predictions point of view would be the gray is the pollen trend advancing. The green is the EVI or vegetation greenness advancing. They both seem to be following each other. They're also following some meteorological variables. But you would think then it's a sure thing that you just take this EVI value here and you know what it's going to be up here. But it doesn't work that way because the interannual variation is crazy. And it turns out that this interannual variation of the satellite greenness is not follow exactly the interannual variation up here. So that's work that we're still doing. But it's no guarantee just because you see this coherence that it's going to allow you to predict next year, since every year seems to be a different climate here in Australia. Sorry about that. Okay, I'll move on to finish up. So to finish, I guess these are just some challenges. This is getting into the world that I do not know, but similar to the OSFlux groups, we have pollen footprints, and we really I don't think that we have been able to characterize the footprint of a pollen sampler site that well, since we'll need to know that if we're ever going to establish a national pollen monitoring forecast system. And the thing about these footprints is that, of course, they change diurnally with wind speed and wind direction, and they'll change daily quite rapidly. And each species has different size, weight, aerodynamic properties. So the footprint will be different for every type of pollen. And all of these, I think, gets into a very complicated but interesting realm. And what we're hoping is that eventually, you can, for any spot, like where the pollen sampler is, you can use wind speed data like his-split or BARA and back trajectories to figure out where the sources might be. And there are were two recent studies that kind of dealt with trying to do such predictions. This one by a colleague in Serbia, Nikovic, that did some sophisticated dispersion models to predict the activity during the thunderstorm asthma. He just used a static grass map and then tried out his predictions. And then there was another paper too about modeling the thunderstorm asthma by Catherine Emerson at CSIRO. And that one is more interesting to me because they actually, I guess there's supposed to be another graph here. So they were actually using satellite data real time, EVI, and they used, rather than the peak, they used the end of the growing season as defined by EVI as a better indicator of pollen. So they're sort of following the EVI methodology, the phenology, and so on, in line with what we're doing as well. So, okay, so I ran over, but thanks.
Thank you, Alfredo. Are there any questions?
I have one, Jamie. Can I go ahead?
Yes, please.
Oh, thanks, Alfredo. Really informative talk, a nice overview of everything that's been happening and how it's been reliant on different data sources and the advances in the modelling. A really, really nice tour. What I was interested in is near the end of your talk, and you showed those really enticing downward slopes into getting earlier in the year for the EVI and the pollen. And you mentioned that the interannual variability meant that you really couldn't just make a simple model of the one to the other. My idea there would be, you also showed us, and we know this, that there are many other pollen types. So imagine that the pollen data, and I wanted your clarification on this, is actually not just grass, but everything. And the EVI is also everything. And I'm just wondering if we had more information on the pollen types that might be lagged. So where you get an early greening, that's because the grasses have gone to reproduction quicker, which gives an opportunity for something else maybe to be in reproduction that's producing pollen.
Right. I mean, yeah, it could be. I mean, we know that the EVI is at least looking at grass.
Yep.
But of course, looking at grass in the Victoria doesn't mean you're looking at ryegrass. There's a lots of native species as well as exotic species. And yeah, you're right. What I wouldn't be able to answer is to what extent the size, mass, and aerodynamic properties of, say, ryegrass differ from a native C3 grass species, which again, like you said, it could cause that type of variation.
Yeah. But we don't have the scale of monitoring to pull apart the pollen types even within the samples yet, do we? Not at scale.
Okay, so it's very hard to identify grass pollen. Light microscopy is used, but all you could do is pull out that it's grass, poisson. So now with metabar coding and DNA analysis and so on, you can actually get down to the genus level and say it's Pooidae or Panicoidae or these. I'm not that aware about to what extent that would affect the wind transport of these. But yeah, maybe each grass type may have a different transition from vegetative to reproductive phenology, and some may instantly or quickly release, and others may wait and lag. So because I would think the exotic ones may not behave the same as native because they grew up somewhere else.
Yeah.
So
it's a can of worms in a way, isn't it? but it's still really important that they just at aggregate, they're all the allergens. And I guess the end point being, people suffering higher asthma attack tells us whether the model's got some good signal in it anyway.
yeah, you're just trying to at least improve people's lives.
Alistair has a question.
Hi, Alfredo. I'm a marine biologist and I kind of work in forecasting in ocean areas. It was interesting to note that the trend you found in the greening phenology, we're also seeing in chlorophyll off the east coast of Australia, where that phytoplankton bloom is happening earlier in the year also. But my question to you is, you've had a look at the greening index versus pollen, and then rainfall versus pollen. What happens when you put rainfall and greening into a predictive model of pollen?
Yeah, I mean, I'm not there yet.
Yeah.
But to what extent, it's hard. I played around with some predictive ML, and I'm not a dispersion modeler. So it's kind of hard, what is going to be the future direction for this. But you're right. I mean, it's good to look at, it's good to take the residuals of that pollen EDI, and from that, try to see if we can back out, or like you said, add some more predictive variables to that and see if that will, to what extent that handles the residuals. But yeah, we haven't done that yet. We barely got the advancing done. So
yeah, I think, you probably got to search that space between different variable combinations, and then also even your metric, as you talked about with Catherine Emerson's work, where she's used a different part of that greening curve. There's a search for which one is the best predictor out of all the possible places you could pick on a curve. Yeah.
Yeah.
Thank you.
No, just because Catherine is here. Catherine is listening to your talk, Alfredo. So yes, we did a full 3D model with meteorology and wind speed and wind direction and all manner of things. So generally, obviously, the rainfall lowers the amount of pollen in the air, but also a high humidity reduces the amount of pollen that's being released at the time. So there's all these different things that come into play in the sort of 3D model. But yeah, good talk, Alfredo.
Thanks. Yeah, it's sort of like, do you try to predict at weekly scales? Do you try to aggregate the high frequency wind data so it's more manageable at weekly, and then see if you can predict the weekly before venturing inside into the daily scales and then even sub-hourly kind of scales? So some of the pollen data is available on hourly. It's just a matter of how much time and effort spent to count the slide that it's collected on, or the drum that's collected on. And people do that for special experiments. But I imagine it's similar to OSFlux people that, they've got a specific purpose and they want to look at the hourly data to figure something out. But we all know that if we're going to combine satellite data with flux data, that more or less currently people are happy with weekly. And now people are curious whether daily will add anything. Like, do you expect chlorophyll? Maybe you expect fluorescence can change day to day, but would chlorophyll really significantly change, or just lag, what the environment is doing? So I see this happening with the same thing with pollen, but spatially and temporally. The scales just are crazy, I think.
Belinda?
I think Alfredo's actually also already answered my question, but I still wanted to say thanks, Alfredo. That was fantastic. That was really super helpful. Sitting here with Jürgen, who's currently developing a model to try and predict C3, C4 grass distribution. We've been talking to Chao Yun a lot and kind of thinking about all the mechanisms that lead up to the patterns in phenology that you're seeing, which we haven't really thought about pollen as being an additional thing to constrain the models. But I guess the question I was going to ask was really around the predictors. Like, what sorts of predictors are you considering, and what sorts of predictors seem to have the most potential for predicting at the moment? I don't know if you have any teasers to give us.
Gosh, not really. I mean, I'm interested in that. It's sort of like the approach right now is I'm just trying to see what can I stretch out of the satellite data. So, like, if we take the grass signal from satellite, to what extent is that grass, living grass, has it already integrated everything we need to know about the soil, everything we need to know about meteorology? Can we trust that grass to tell us the history, at that moment? Or maybe not. So, or maybe the satellite has its own artifacts that's masking or giving us, the wrong message or confusing message. But other than that, it's just like playing around a little bit with machine learning. We just start adding precipitation and temperature, and I don't know to what extent we can just add it like a garbage can, or do you have to tease out lags, or does it handle the lags by itself? So that's kind of like what I tried to say to Jamie when I first got the invitation for the talk, that I haven't delved deeply into actually predicting, but I'm trying to find ways that I think will work.
Yeah.
And
cool. Thank you. It was really great to hear you talk about the ACS workshops as well. That was so, spectacular what's come out of that workshop. Just reflecting on how productive that was, and sitting here with the younger generation going, yeah, God, we need those workshops back again.
Oh yeah, those were great to get people actually in the room together, right?
Yep.
From different backgrounds. And it was good timing, because little did we know that there was a thunderstorm asthma event coming. So all of a sudden it became in the news everywhere, and very hot topic to study.
Glenda?
Yeah, just following up, Alfredo, and to where your strengths are, I know. Has anyone in remote sensing thought about near-term forecasting of the actual EVI values themselves? We're always using it as the tool to get to somewhere else, but surely the processes generating that now are a bit better understood, and it would be quite useful as a product to have to put into biological models.
Mm-hmm. Yeah, it's been on my list to do, especially like when I heard about the NEON or the challenge, about, can you tell us what the phenocam value is a few days from now, or that type of prediction. And I know that this one guy, I guess Dick Waring, I don't know if you know, he's in Oregon, is it? Okay. But yeah, he was like, he's fascinated by the study where done by Kazuhito Ichi in Japan, that he basically predicted the rooting depth of Amazon forests using EVI.
Oh.
Because it's basically his model had all the parameters in it. And if he trusted the vegetation index and he adjusted his model by just keep increasing rooting depth, to predict EVI when it actually matched the EVI is how he established his root depth map. And that's what he published in GRL. And then I was told by Dick Rarian, that's exactly what you should be doing. You know, use the EVI as a predictor for something else. So it's in my mind, but gosh.
Yeah, we need the equivalent of ACES and opportunities that allow teams to come together and work at depth, right?
Yeah.
I think so.
So it's sort of like using the data for what it is rather than trying to force it
Yes.
to do what you want it to do. Or
Yes.
yeah, it's kind of fun to try and play around that way.
All right. If there are no further questions, I'd like to thank Alfredo one more time for giving us his time.